solve that problem. So obviously, if you turn it in late after I've already gone through the answer, it's pointless to do that. So I need you to turn it in on Friday during class or earlier. You can email it to me if you like. So G proteins represent sort of a segue to what we talked about last week. So last week we talked about receptors. Remember that Paul Ehrlich, the father of pharmacology, defined a receptor as being two components: a binding site, which is the site that binds the drug and is the recognition site, determines the affinity of the drug, and it determines the specificity of the pharmacological response. But the effector is the second part of the receptor, and that is the component of the receptor that translates the binding of an agonist into a biological response, in this case an intracellular response. So G proteins represent an example of an effector system. As a matter of fact, for G protein coupled receptors, it's the G protein that serves as the intermediary, the middle component. People call it the transducing element, the element that connects the receptor with the effector. So today's lecture, I'm going to focus in on G protein structure and function, and then on Friday I'll talk about the receptors and the effectors. Okay, so that's how this works. I want to spend a couple of minutes going over the two major components, the two major types of receptors that you see in the nervous system, and actually in a variety of other peripheral systems as well. And we can compare and contrast these things. So the nervous system consists primarily, there are other receptor superfamilies as well, but the two biggest superfamilies in terms of the types of receptors we have are the ion channel receptors, also called ionophoric receptors, and the G protein coupled receptors. So a classic example of the oligomeric ion channel receptors are the cholinergic nicotinic receptors, which is of course part of your Homer problem. Also, we talked about it last week as well. So that's the receptor not only present in brain that binds to acetylcholine and nicotine, but also clearly the receptor involved in contraction of muscle. But a variety of other receptors important in the brain. For example, the GABA-A receptor is the predominant inhibitory receptor in the brain. The glutamatergic receptor is a primary excitatory receptor. So these ionophoric receptors are an oligomeric association of different protein subunits that form a complex that creates an ion channel across the membrane. The function of that ion channel, whether it transports negative ions or positive ions, really depends on the receptor that you're looking at. So you can't really separate the receptor binding site from the channel. All of these proteins form an oligomer type structure, very complicated structure. And of course the signal transduction mechanism here is to open an ion channel. The G protein coupled receptors are quite different. The classic examples of these are, for example, the beta adrenergic receptor, which is the predominant receptor in the autonomic sympathetic nervous system, but also in brain as well. Acetylcholine not only binds to ionophoric receptors, but also binds to muscarinic G protein coupled receptors. The function of the G protein coupled receptor is to form a ternary complex with a G protein, which is a protein that binds GTP or guanosine triphosphate, and that G protein forms a transducer that connects the receptor with the effector. So that's called a ternary complex because you've got the receptor, the G protein, and the effector. These are different proteins. So unlike the ionophoric receptor, where all of these protein subunits form one gigantic complex, the G protein ternary complex are separate proteins that collide and interact with one another in very specific ways. And the mechanism is to activate a variety of second messengers. The most well studied of these is cyclic AMP, but there are also other effectors like phosphoinositol and other ion channels that I'll talk about on Friday. These two superfamilies of receptors have some similarities and some differences. The main difference, in addition to their structural properties, which are very different, is the speed of response and termination. So ionophoric receptors are built to respond very quickly. They bind to their agonist very quickly, and they terminate very quickly. So their function is quick on and quick off. On the other hand, G-protein coupled receptors have long durations of action and delayed onset, okay? So these receptors don't normally activate as quickly as the ionophoric receptors, and they proceed for a much longer period of time. The length of the duration of that action depends upon the type of effector that we're looking at. So this really is sort of the nutshell of how a nervous system works. The nervous system has to do a lot of different things. Partly it has to respond very quickly to its environment, but at the same time it has to have the capability of having slower actions and longer acting actions in order to work. So both of these kinds of functions are the reason why we have these two superfamilies of receptors. But for both superfamilies, really the function of the receptor is to act as a catalyst for their agonist, whether it's a hormone or a neurotransmitter. And the way that works is in every case there's a point of amplification. The binding of one molecule of agonist, whether it's a drug agonist or a neurotransmitter, produces an amplification so that the signal response inside the cell is much greater than you would have predicted by simply binding one molecule of agonist. So in the case of an ionophoric receptor, the amplification comes from the fact that you've got all of these ions that are coming in to create a change in the excitability of the cell. In the case of G-protein coupled receptors, you have all of these different signal transduction steps. They're very complicated, but they occur one after another, and there's amplification at each step along the way. So that if there's a tenfold, I'm just making up a number, if there's a tenfold amplification along 5 different steps of a receptor, all of a sudden at the end of those 5 steps, you've got a major big response occurring inside the cell. And I'll show you some examples of that amplification on Friday. There are all kinds of different levels of complexity, the way in which the effector and receptor interact with one another, and I won't talk about that very much for the ionophoric receptors. And regulation is very different in these two superfamilies. We talked about the regulation, desensitization of nicotinic receptors last time. I'll talk about the desensitization of this family of receptors on Friday. Here's a little history, very, very interesting history with some very colorful personalities that discovered G-proteins and G-protein coupled receptors. A lot of important people, many of whom won Nobel Prizes. This field really began by Earl Sutherland back in the 1950s. And he did more than just discover these kinds of receptors. He really created the concept of signal transduction. So before he came on the scene, people thought that signal transduction, the ability of a receptor to produce a response inside the cell, was a black box. It really was so complicated that no one could ever understand it. And in fact, the signal transduction had to occur within an intact cell. There was no way for you to take the cell apart and examine biochemically the components that were going on, because the cell was itself required for the signal. But he showed that you didn't need an intact cell. He was working with liver, and what he did is he threw a liver in a homogenizer, a blender, and he broke everything up. And he found that you could still get a receptor response. The receptor he was looking at was the beta-adrenergic receptor. Epinephrine, or norepinephrine, was the agonist for that receptor. And he found that when he added epinephrine to these membranes that he isolated from liver, he got the formation of a small heat-stable molecule that was present inside cells. And he found out later that that was cyclic AMP. And so this was the response that you could see, not only in an intact cell, but just in a test tube. And so he was the one that discovered that the beta-adrenergic receptor was coupled to cyclic AMP and stimulated cyclic AMP in the cells. And so he called cyclic AMP a second messenger. He really derived that term because he viewed the process of signal transduction as occurring in two steps. The first messenger was the receptor itself, and the second messenger was the formation of an intracellular diffusible compound that could go with, inside the cell and create all kinds of signals. Sutherland was a very interesting character. At a very early age, he became an alcoholic, and was an alcoholic throughout the time he made these great discoveries. People who worked with him knew that you had to get to him very early in the morning before he started usually drinking about 10:30, 11 o'clock in the morning, and by 3 he was incoherent. But in those 2 or 3 hours every day, he was brilliant and really solved these problems. Won the Nobel Prize, I believe, in 1962, about 6 months before he died of cirrhosis. So he just barely made it under the wire. The next phase of the operation, this guy here, Gordon Tompkins, is an unsung hero. He was a geneticist, and he was the one who began to figure out that not only was there 2 messengers involved. Sutherland said there was a first messenger and a second messenger, but by using genetics, he was the one who figured out that there's a third component as well, that there is some kind of a protein in there that's required to couple the beta adrenergic receptor with adenylate cyclase, the enzyme that forms cyclic-AMP. But Tompkins died in a car wreck before he could identify what that was. So he was at the University of Virginia in Charlottesville, and so one of his faculty colleagues in Charlottesville was Al Gilman, and he took his cell models that he used, and he was able to show that the third protein, that central component that coupled the receptor with cyclase, was a G protein. And another guy at NIH, Martin Rodbell, did exactly the same thing. So Rodbell and Gilman shared the Nobel Prize. And then down the road at Duke, Robert Lefkowitz was the first to clone the beta adrenergic receptor and show its similarity to rhodopsin, and I'll talk about that in a minute. And then just a couple of years ago, Brian Kabilka solved the structure of the beta adrenergic receptor with X-ray crystallography. So it's a long and profitable history. So here's what the ternary complex looks like. Once again, these are separate proteins. They are all plasma membrane proteins. They can interact with one another or dissociate with one another. So the receptor, the G protein receptor, is a single subunit protein. It contains the binding site for the agonist and the antagonist. Okay, so this, once again, is the recognition site for this receptor system. The effector represents a variety of all kinds of different enzymes and channels. These are, this particular slide is for the subclass of receptors that couple to the inhibitory GI and GO proteins. So the effectors for these is inhibition of cyclic AMP, inhibition of calcium, and stimulation of potassium, but there are other effectors as well. But the thing that glues them all together is this G protein, okay, which has 3 subunits involved in it, alpha, beta, and gamma, all of which are very important in this whole process. I'll define it in a minute. And the function of this is to create kind of a glue, an intermediary, and really it's called the transducing element, which allows the receptor to couple to the effector, and it does so by binding GTP, or guanosine triphosphate. So GTP is a required element of this entire process. There is no energy directly required for the coupling of receptors to effectors, but GTP is hydrolyzed to GDP, not to create energy, but this is the termination step, and we'll see this a little bit later on. When the protein binds GTP, it becomes activated, and when the GTP is hydrolyzed to guanosine diphosphate with the release of phosphate, that inactivates the whole process. We'll talk about exactly how this works in just a couple of minutes. Okay, so let's take a look at these subunits that form the basis of the signal transducing element, the G protein. And of course, there are 3 of them, alpha, beta, and gamma. But we really only talk about 2 because we talk about alpha as a separate subunit, and then we talk about beta gamma together because beta gamma are two subunits that are always together. They're always they never really fall apart, and so we talk about them as sort of one. The alpha subunit is what I'm going to talk about primarily, because that represents primarily the business end of the G protein. So the G alpha has a number of important functions. The alpha binding site, the alpha subunit of the G protein, first of all, contains the binding site for guanosine triphosphate. And that is the binding of GTP is the signal that activates the G protein and allows the receptor and the effector to interact and produce an intracellular response. So this is the activation site for the entire process. But it also has an intrinsic enzyme called GTPase. Okay, so the function of GTPase is to take the guanosine triphosphate, remove the terminal phosphate, and produce GDP, guanosine diphosphate. When you do that, that inactivates the G protein. It inactivates the ability of the receptor to produce an activation of the effector, and therefore it terminates the activity of the entire system. And finally, G alpha is the binding sites and the functional sites for two very important bacterial toxins. Cholera toxin, of course, responsible for cholera, which is involved, which is a toxin that binds to the GTPase on one of the subfamilies of G alpha, and pertussis toxin, which is binding to another subclass of G protein called GI and GO. And I'll talk about these toxins in more detail a little bit later on. So, the identity of the G alpha subunit determines which effector is going to be used. So there's all kinds of effectors that mediate the biological functions of these receptors. And it's the identity of the G alpha that determines which effector is going to be activated or inactivated depending upon the type of receptor you're looking at. So there are 5 major classes of G alpha. There are also some minor classes as well, which we're not going to talk about today. The first one that I'll talk about is G sub T, okay, which is also called transducin. It's a very specialized G alpha subunit. It's found entirely within the rod outer segment of the retina, and it's involved in transducing the signal of light into a biological response. The effector in this system in the rod outer segment is an enzyme known as cyclic GMP phosphodiesterase. And we'll talk more about all of these effectors on Friday. So transducin is really very specialized, only really present, as far as I know, in the rod outer segment. The rest of these, the other 4, are G proteins that exist throughout the nervous system and as well as in peripheral tissues that mediate hormonal actions. The best studied of these would be GS, the stimulatory G alpha subunit. This is the system that was originally described by Earl Sutherland, although he had no idea at that point that there was a G protein involved in that. So the function of GS is to stimulate an adenylyl cyclase. This increases cyclic AMP in the cell. Cyclic AMP then diffuses inside the cell and activates protein phosphorylation through an enzyme known as protein kinase A. Okay, so phosphorylation of these proteins within the cell by this GS-mediated phenomenon changes the activity of a whole variety of different enzymes in the cell. GI is the inhibitory G protein, and its function is exactly the opposite of that of GS. It binds to adenylyl cyclase, but instead of activating it, it inhibits this enzyme, so it produces a decrease in cyclic AMP inside the cell. So that, of course, leads to a decrease in protein phosphorylation, and thereby counteracts the effects of stimulatory receptors that increase cyclic AMP. GI also has another function. GI interacts with potassium channels on nerve cells, and it opens potassium channels. It's called an inwardly rectifying potassium channel. I'm not going to go into detail about that. But when you open a potassium channel in a nerve cell, this particular potassium channel in a nerve cell, you produce hyperpolarization. That's an inhibition of nerve cell activity. The hyperpolarization essentially means that it reduces the probability that that nerve cell is going to fire. So it inhibits nerve cell firing activity. So when you think about GI, it's a purely inhibitory function. Okay? It inhibits in a whole variety of ways. Very closely associated with GI, very closely related to GI is GO. GO is also an inhibitory G protein. It inhibits adenylate cyclase just like GI does. It also has another function in brain, very important, and that is to inhibit calcium channels. Now, calcium is a critical ion for the proper functioning of nerve cells. Calcium is required for a whole variety of things in nerve cells, but most importantly, calcium is involved in the release of neurotransmitters from the presynaptic cell. So when you release a neurotransmitter from a presynaptic neuron, the way this works is that you get an influx. When the action potential propagates down the nerve cell, there's an influx of calcium into the cell, and it's that calcium that represents the signal for the release of neurotransmitter into the synapse. Receptors that couple to GO close these calcium channels, prevent the influx of calcium into the cell, and by doing so, they block neurotransmitter release. So these kinds of receptors, a very common example of these kinds of receptors are called presynaptic autoreceptors. These are receptors which are designed to inhibit their own neurotransmitter release. Great example of this in the adrenergic system is the alpha-2 adrenergic receptor. So remember, adrenergic receptors are receptors that bind to norepinephrine. Norepinephrine binds to the beta receptor to stimulate cyclic AMP, but presynaptically norepinephrine can bind to the alpha-2 receptor to inhibit its own release. It's a negative feedback mechanism. Very important mechanism in brain, and you see a lot of these receptors around. So when you hear about presynaptic receptors, that's a lot of what they're talking about. And finally, GQ, some people call it GP. So you see that both ways, and I in fact may even list it both ways because I'm neutral on that controversy. Is a stimulatory G protein activity. Okay, so GQ activated coupled receptors stimulate the turnover of a lipid in membranes called phosphoinositol. And what this does is it increases calcium into the cell. Okay, so it does exactly the opposite of what GO does. And it also increases the activity of a protein kinase called protein kinase C. So both of those actions produce excitability, produce an increase in activity of the neurons. So this is a stimulatory G protein. So if you want to look at it in terms of functions, we have 1, 2, 3 subfamilies of G alpha that are stimulatory and 2 that are inhibitory. This is a complicated cartoon, and I want you to ignore all of this stuff down here. Let's just look up here. This cartoon really represents a map of the structure of a G alpha, and it shows you, where on the protein different things are occurring. So the G alpha, it's not a big protein. It's what, 35 kilodaltons, but it's got to do a lot of things. It has a lot of things going on here. So this sort of represents the two-dimensional view of the amino acid sequence of a typical G alpha. So here's the amino terminal end, and here's the carboxy terminal end. And of course, these proteins are not linear. They're all folded up in a very specific way. So the GTP binding sites are represented here, and it's not that these sites are are different from one another. There's just one GTP binding site, but if you imagine this line here sort of folded up in a very complicated three-dimensional structure, all of these GTP binding sites fold together to create one binding site. So that's not only the binding site for GTP, but it also represents the enzyme GTPase, which terminates the G protein function. On the C-terminal end, the G-alpha subunit binds to the receptor. So that's the portion of the transducing element that couples to the receptor, allows the receptor to do its thing. And then on the totally the opposite end of the G-alpha subunit, you have the ability of the G-alpha subunit to bind to beta gamma, which is critical for its function that I'll talk to you about in a few minutes. And we also know exactly where these two bacterial toxins work. Cholera toxin works right here on an amino acid that's critical in the GTP binding site, and pertussis toxin acts right here on an amino acid that's critical for the binding of the G-alpha to the receptor. And I'll show you the consequences of that in just a minute. So we do not yet have a fully worked out X-ray crystallographic structure of G-alpha, but there are various bacterial homologs of G-alpha that we have that we can figure out what's going on. So this is, obviously I'm not going to hold you responsible for any of this, but this just simply represents a cartoon representation of how people think of G-alpha and how it folds up, and which amino acids are involved in binding to GTP and other sorts of things. There are subfamilies of G-alpha. We don't know much about these, but you will run into them in the literature. So here is an example of different subfamilies of G-alpha subunits that interact with the D2 receptor. The D2 dopamine receptor is an inhibitory receptor, and it binds to G-alpha and GO. I'm sorry, GI and GO. Well, so it turns out that there are three subtypes of GI that the D2 receptor works in. GI1, GI2, GI3, and all of these do pretty much the same thing. They inhibit cyclic AMP. There's a GO that decreases calcium. There's a GZ, which is another subfamily that I won't talk about today, and other things involved as well. So we don't really know why, for example, there are three subtypes of GI around. There are two subtypes of GS. They all seem to do the same thing, but they have slight differences. So there must be some reason why these subfamilies of G-alpha exist, but we're not exactly sure why. So remember that I said that the identity of the G-alpha determines the identity of the effector. I'm also going to tell you that the identity of the G-alpha also determines the receptor as well. And the rule is that a receptor will only bind to one type of G-alpha, period. So rhodopsin is the receptor in the outer rod, the rod outer segment of the retina. The rhodopsin is the protein that is activated by light. In the case of the rod outer segment, the agonist for this receptor is a photon of light. Okay, it's unusual for that reason. Rhodopsin does not bind to any other G-alpha other than transducin GCT. Beta adrenergic receptors is a classical stimulatory receptor. Once again, it binds to GS. It does not bind to anything else. Opioid, there's a whole series of inhibitory receptors in brain that bind to both GI and GO, and then serotonin 2 receptors bind to GQ. So once again, these receptors only bind to one subtype of G protein, alpha subunit. You can mutate receptors to bind to other types. So people have taken the beta adrenergic receptor and mutated parts of it in ways that allow it to bind to GI, but that's a trick. That's not a natural phenomenon. The other thing this tells us is that receptors exist in multiple subtypes, and this is not just a specialized kind of thing. This is a general rule that exists in virtually every case of neurotransmitter that I'm aware of. If there's something that violates that rule, I'm not sure I know about it. So, norepinephrine, for example, which is misspelled there, but it's not supposed to be norepinephrine, but norepinephrine, right? So it binds to the beta adrenergic receptor to stimulate cyclic AMP through GS. It binds to the alpha-1 adrenergic receptor to stimulate phosphoinositol turnover through GP or GQ. And it binds to the alpha-2 adrenergic receptor as a presynaptic autoreceptor. So, norepinephrine can be either a stimulatory neurotransmitter or an inhibitory neurotransmitter, depending upon which receptor we're talking about. Cholinergic system is even more complicated because in the case of acetylcholine, there are actually 4 families of G protein coupled receptors, M1 through M4, but here's 2 of them. M1 muscarinics are stimulatory, acting through GP or GQ. M2 muscarinics are inhibitory, acting through GI and GO. But acetylcholine not only binds to these muscarinic cholinergic receptors in the G protein superfamily, but they also obviously, acetylcholine obviously also binds to the ionophoric receptor, the nicotinic receptor. So acetylcholine is even more complicated. The most complicated neurotransmitter system of all, in my opinion, is serotonin 5-HT. I think there are 7 or 9 subtypes of serotonin receptors. I can't keep up with them all, so I'm not exactly sure. The serotonin 1A, for example, is an inhibitory receptor. The 1C is a stimulatory receptor. The 2 subclass is a excitatory receptor, GP or GQ. There's a 5-HT3 receptor that's an ionophoric receptor, like the nicotinic receptor. So serotonin has a very complicated pharmacology. So what does this mean from a pharmacological point of view? This is really critical because it tells us that there are multiple receptors that mediate the function of every neurotransmitter in the brain. And from a pharmaceutical point of view, this is a massive development because it suggests that we can develop drugs that are specific to each subclass of receptor. So we can, in the adrenergic field, we can provide beta adrenergic antagonists, which are antihypertensive. They act to reduce blood pressure and heart rate. You can have alpha-1 agonists that treat asthma. You can have alpha-2 agonists that treat hypertension, okay? So all of these, the entire set of pharmacology that we have today is all based upon the principle that there are multiple functions for each of these receptors, and we can define those by working through different G protein alpha sites. Okay, so that gives you a survey of how these G proteins are, what they're doing. Now we're going to go into an extended discussion of exactly how this works from a mechanistic point of view. This slide here is kind of a summary. I'm going to go through it very quickly because I'm going to come back to all of these points in more detail. So, the G protein family of receptors exist, as we know, as a ternary complex, a complex between the receptor, the G protein, and the effector. In the basal state, the G protein has a high affinity for GDP, guanosine diphosphate, a low affinity for GDP. So in this state, the G protein is inactive. When the receptor is activated by agonist, it produces a change in the conformation of the G protein alpha subunit so that it now has a high affinity for GDP and a low affinity for GDP. And because there's a low affinity here, the GDP dissociates very quickly. at the receptor at the receptor level in the basal state, in the inactive state, there's a bound complex between the receptor and the G protein, and the G protein in this state is a heterotrimer where the alpha, beta, and gamma subunits all fit together. And when the receptor is activated, all of these things fall apart. The alpha subunit and the beta gamma subunits fall apart, and the receptor falls apart from the G alpha. And because of this activation, you see an increase in GDPase activity and GDP release. So now let's go through this scheme here and show you exactly what this means and how it works. So this is the G protein activation cycle. And it shows you the change in what's happening in every component in the G protein ternary complex, including the receptor, the G protein transducer, and the effector. So I'm going to start up here, okay? This is the basal state, or the inactive state of the receptor complex. So in this state, we have the receptor, which is bound to the G protein trimer, alpha, beta, gamma, and in this state, the G alpha subunit is bound to GDP. We also have a scenario, this is the receptor I've listed as R star, and what that means is the receptor in this particular state has a high affinity for the agonist, okay? High affinity for agonist. Now, because it has a high affinity for agonist, when agonist comes on board either as a drug that we administer or a neurotransmitter or hormone that's released from a presynaptic cell, the agonist has a high affinity for the receptor, so it has a high probability that it's going to bind. And when the agonist binds to this complex, it produces a change in the conformation in both the receptor and the G protein. And when that happens, the G protein switches over into a low affinity state for GDP. Now remember what I told you last week about the relationship between affinity and dissociation rate. Remember that low affinity states mean rapid dissociation. So when the G protein is in its high affinity state for GDP, GDP is stuck on there. It doesn't come off quickly. But when it's switched into its active form, GDP switches over to a low affinity state and it dissociates, it's gone. And when GDP dissociates, it's released from the G protein, GTP, guanosine triphosphate, comes on board. So you have this switch between GDP and GTP, and when that happens, everything falls apart. The receptor comes apart, and the alpha subunit dissociates from both the receptor and the beta gamma subunit. So what we have as a result of this activation process is two things. We have the naked receptor, and I'll come back to that in a minute, but we also have these two dissociated G protein subunits. We have the alpha subunit, which is now bound with a high affinity to GTP, and I put a star on that because that indicates that the alpha subunit is now activated. And we have the free naked beta gamma subunits. Both of these two things can then bind to an effector and produce its effects, either activation or inhibition, depending upon the type of effector we're looking at. So some effectors will bind directly with the activated G alpha, and some effectors will bind to the naked beta gamma. Now, no effector will bind to alpha beta gamma when they're together. It can only bind, and they can only be activated or inhibited only if these two subunits have been dissociated from one another. So both the activated form of G alpha and beta gamma are transducers. They activate or inhibit the effector. All right, so I'll get back to the receptor in just a minute. Now remember that in the nervous system or in any cellular response, it's critical not only to have an activation phase, but it's also critical to have a termination phase. Now GTP, okay, is bound with high affinity to the activated G alpha. So it's not going to dissociate very quickly. And so that's not fast enough for this process to really terminate. So the G alpha subunit has designed an intrinsic GTPase which takes GTP and removes off the terminal phosphate and provides with a GDP, okay? Now, once that hydrolysis occurs, this activated G-alpha now becomes a GDP inactivated G-alpha, okay? And so this inactivated form of G-alpha can no longer do anything to the effector anymore, and all it can do is to find a free molecule of beta gamma to bind to regenerate its own inactive complex. But let's now talk about the receptor. Okay, now the receptor has, once it's been activated by the agonist, the receptor has dissociated away from the G protein subunits. When that happens, when this dissociation reaction occurs, the receptor shifts from the high affinity state into a low affinity state for agonist. And when it has a low affinity for the agonist dissociates. It's gone. All right? And now that it's the receptor is now low affinity state, it's naked, it's not bound to agonist, it's not bound to anything anymore. It now has the ability to bind to the GDP form of alpha beta gamma and regenerate the basal complex. When you do that, when these two things come together, the G protein trimer and the receptor, you now regenerate the high affinity form of the receptor, and this whole complex is now ready to start all over again for a new round of stimulation. So notice the sort of the beauty of how this thing is involved. Receptor is in its high affinity state in the inactive form. That allows it to be like a sponge for agonist, okay? And anytime an agonist is going to come out into the synaptic cleft, this high affinity receptor is going to bind to it, okay? Because it's present in its high affinity state. But once the agonist has done its job, you don't want the agonist around anymore. If the receptor just stayed in its high affinity state, the receptor would stay activated for far too long, okay? And that would be bad news. You've got to have the ability to regenerate this cycle over and over again, all right? So the fact that once this process of activation has occurred and the receptor has dissociated away from the G protein, now it's in its low affinity state, that allows the receptor to dissociate, and now you have the ability to regenerate the basal complex and have a new round of stimulation. So all of these changes in affinity states are all there for a reason, okay? They're not just random events. They occur because it's the way in which this system works to provide the ability for the cell to bind to agonists very quickly and to respond by dissociating from agonists and doing it as well. And so the G protein does the same thing. You have these different affinity states for G-alpha and guanosine, triphosphate, and GDP. You also have different affinity states for G-alpha versus G-beta, and you have different affinity states for G-alpha and the receptor. All of these different affinity states, high affinity states, low affinity states, are also involved in the dissociation process, all right? So as long as G-alpha has a high affinity for the receptor, these things stay bound. But when G-alpha is activated by GTP, it now is in a state where it has a low affinity binding for the receptor, and they fall apart. All of these things, once again, is key to understanding is the ability to know that every time you get a change in affinity state, you get a change in the dissociation rate of whatever component you're looking at, whether it's the agonist, whether it's GTP or GDP, whether it's two different proteins working, with one another. All of these things come together to create this response. Yeah.
At what point is the receptor endocytosed into the cell? Is it right when the agonist binds to it?
I'll go into that Friday in great detail, but the answer is it's endocytosed when it's chronically bound to the agonist. Okay, so this whole process occurs when the agonist dissociates, but when there's so much agonist involved that it stays activated, then you undergo desensitization. But we'll talk about that in great detail on Friday. So this, once again, is a summary slide that tells you exactly what I just said. Basal state is the bound complex, high affinity state for agonist, rapid association, slow dissociation. Activated state is the agonist bound receptor. You dissociate the receptor in the G protein trimer. You get a low affinity state for agonist, fast dissociation rate of agonist. And I'll skip number 3. Critical to this is GTPase. Okay, so GTPase, of course, is the component of the G alpha subunit that regenerates the basal complex, so that hydrolyzes GTP to GDP and terminates the entire signal. GTPase, in its normal form, is a relatively slow enzyme. Okay, it is it's not super slow, but it's relatively slow. And for some effectors, that's fine. Okay, so for example, most types of adenylyl cyclase, cyclic AMP is a long duration effector, so you don't really need for this system to be terminated very quickly. But other effectors like calcium channels and potassium channels, this normal rate of GTPase is just too slow. It just doesn't occur fast enough for them. So they have created a mechanism to speed up this whole process. And some effectors have associated with them what's called GAP proteins. Okay, GAP are called GTPase activating proteins, and the function of GAP is to increase the rate of GTPase activity. So you can imagine here, okay, if we have a GTPase activity that's where the rate is increased, that means that the time in which this G protein activated G alpha subunit remains activated is decreased. Okay, the faster this GTPase enzyme proceeds, the quicker this activated G protein is going to be inactivated into its basal state. So the function of GAP proteins is to terminate this reaction even faster. And when you do that, this whole cycle goes along a lot faster, and that's a good thing for ion channels like calcium channels and potassium channels. So these GAP proteins are well studied. There's a subclass of GAP proteins called RGS. I forget what that stands for, something signaling system. But once again, they're all involved in doing that. So a lot of these, there's a lot of research going in on these RGS proteins because they are modulating the effects of agonist in this system. So the idea is if we had drugs that affected these RGS proteins, we could regulate this system in a very specific way. So there's a lot of interest in these things. And of course, I've talked about this low affinity agonist binding state already, so I don't need to go into that again. Okay, so this explains why an agonist goes through this cycle and activates the effector or inactivates the effector depending on what we're talking about. Now let's talk about these two bacteria. Cholera toxin. So cholera toxin, the cholera toxin obviously produces cholera, a nasty business. Cholera toxin consists of two subunits, A and B. Subunit A is, I call it like a hypodermic needle. Subunit A doesn't really do anything on its own, except what it does is it binds to a sugar residue called GM1 ganglioside on the cell surface. And when it does that, it basically punches a hole in the membrane of the cell, and it allows subunit B to get into the cell. If it wasn't for subunit A, subunit B wouldn't be able to do anything. Okay. So once subunit B gets into the cell, it goes into the cell, and it binds specifically to the alpha subunit of GS. And what it does is this is specific on a specific amino acid that's a critical part of the GTP binding site, but most particularly a critical part of the GTPase on GS alpha. So when the B subunit cholera toxin comes on board, it basically inactivates the GTPase activity in GS alpha. So that means that this process here does not work anymore. In the presence of cholera toxin, this cycle is permanently activated. Here's the reaction itself, okay? And for those of you who, and I'm not going to hold you responsible for knowing this reaction, but basically subunit B is an enzyme, okay? It catalyzes the reaction of nicotinamide adenine dinucleotide and ATP. ATP basically provides energy for this. And what this enzyme does is it takes an ADP moiety out of NAD and moves it over to GS alpha. I don't know where these little squares come from. But basically, oh yeah, I know, it's supposed to be plus, it doesn't matter. Anyway, when this ADP is bound to the GS alpha, it's called an ADP ribosylated GS alpha, and under these conditions, that's what produces the inactivation of the GTPase. So this is the reaction that occurs. It is essentially irreversible because once GS alpha is ADP ribosylated, it can't go back again. So the cell basically, in order to generate new, new GS alpha that's not been modified, it has to synthesize new protein. So people use this in the lab all the time because this is a very nice way to label GS alpha. You can take P32 labeled NAD and throw it in with cholera toxin subunit B, and you can create a radiolabeled P32 radiolabeled GS alpha. So a lot of good research was done with this. When you work with cholera toxin in the laboratory, you generally only work with the subunit B. It's pretty safe when you just have subunit B because without subunit A, it can't get into your cells. Okay, so you don't want to work with the cholera toxin altogether. Now, it's interesting to note how this works from a clinical point of view. We know a lot about this process. So obviously, cholera is a disease of kidney. You know, people basically in cholera, people die of dehydration. They just can't get enough water. And the reason why they're dying of dehydration is because their kidney is not resorbing water. About 95% of all the water that you take in is resorbed by the kidney proximal tubule system. So every time you're drinking a glass of water, it goes out to the kidney, but 95% of it goes back into the body through this resorption system. There is a GS-activated receptor in the proximal tubule that mediates this phenomenon, and when cholera toxin produces this reaction in the kidney proximal tubule, water resorption simply does not occur. So instead of resorbing 95% of your water that you're taking in, now you're resorbing virtually nothing, okay? So in this particular scenario, patients die of dehydration because they simply can't get enough water. And once again, because this is an irreversible reaction, you can obviously have antibiotics that'll kill the bacteria, and that's fine, but you still got to keep treating the patient even after the bacteria are gone because it takes some time for the cells in the proximal tubule to regenerate new GS alpha so that it becomes functional. So that's a problem. So once again, this cartoon tells us exactly where this is functioning right here at the GTPase. Now pertussis toxin is very different. Pertussis, of course, is whooping cough. In terms of its activity, it's actually very analogous to cholera toxin because once again we have these two subunits, A and B. The A subunit punches a hole in the membrane, and subunit B is an enzyme that produces ADP ribosylation. So it looks really similar to cholera toxin except for its specificity, because in this case, the enzyme involved in pertussis toxin does not act at GS alpha, it works at GI and GO alpha, okay? And instead of working on the GTP site, of the G-alpha subunit. It works on the site of the G-alpha subunit that binds to the receptor, and specifically the portion of the G-alpha that's activated by the receptor when the receptor binds agonist. So once again, here is the reaction for pertussis toxin B. You can see it's exactly the same as the reaction for GS-alpha. The only difference is that now we've got GI and GO alpha subunits being involved. So what happens is that when G-alpha, I'm sorry, when the, let me get back to this other little cartoon here because it makes it easier for me. So here's pertussis toxin, there's cholera. Pertussis toxin acts here, and what happens is that when it binds to a specific amino acid, the receptor can no longer activate the G-alpha, okay? So the receptor itself is fine. The receptor binds to the agonist produces a conformational change in the receptor, and that conformational change would normally be enough to activate the G-alpha, but it doesn't happen, okay? So the G-alpha for GO and GI is virtually inactivated. It cannot be activated by agonists anymore. So what we see, come back to here, is the G protein cycle stops right here, okay? Agonist is binding to the high affinity state of the receptor, everything looks good, but then bam, everything's gone. So this cycle is over with, there is no further activation that occurs. So prevents the activation. It, once again, like pertussis toxin, it is irreversible. And it is responsible for whooping cough. To be honest, I don't know exactly how. I don't know what cells in the lung are really critical for this, but I'm sure there are. So notice that these two bacterial toxins are doing diametrically opposed things to the G protein. In the case of cholera toxin, you're getting an irreversible activation of the cycle. In the case of pertussis toxin, you're getting an irreversible inactivation. But of course, the two G proteins you're looking at in each case are different. One is GS and one is GI or GO. Okay. What about beta gamma? We haven't talked much about them. We've talked mainly about alpha, and I'm not going to spend much time on them. Okay. Beta gamma, and once again, we really refer to them together, not separately, because they really do seem to act together. They don't dissociate from one another like G alpha does. If they beta gamma stays together. There are multiple subtypes of beta gammas. There are at least 4 different genes that code for betas and 5 that code for gammas, but they all seem to work the same. I don't really know why they have this heterogeneity. So they do 2 things. Actually, 3 things. It is required, beta gamma subunits are required for G-alpha activation. First of all, these are very lipophilic proteins, so they anchor G-alpha in the membrane. So when the alpha subunit is bound as a complex with beta gamma, it's just sitting there in the membrane. When the alpha subunit dissociates from beta gamma, the alpha subunit is free to go around all over the place. It can go inside the membrane, or it can be released from the membrane inside into the cell. But beta gamma stays in the membrane, so it's an anchor for this whole process. And when it binds to G-alpha, it stabilizes GDP binding. Okay, so once again, as we talked about before in the G protein cycle, when beta gamma is complex with the alpha subunit has a stable high affinity for GDP. And that once again produces that basal state that is so sensitive to agonist, because that produces a high affinity state for the receptor. So once again, beta gamma, very important for the G-alpha activation process. But beta gamma also has a very important role on its own, because when beta gamma is released from the activated form of G-alpha, it directly modulates some effectors. Some subtypes of adenylyl cyclase are activated by beta gamma. And ion channels seem to like beta gamma as well. Okay, so beta gamma is a direct modulator of effectors, the free beta gamma. The bound form of beta gamma, when beta gamma is bound to alpha, doesn't do anything to the effector. Pharmacologically, we can measure the function of G proteins by a radioligand. So this is a radioligand. Once again, last week we talked about radioligand binding for receptors. Well, you can also do radioligand binding for the G proteins. And what you use is an S35 labeled molecule called GTP gamma S. Okay, so here is GTP itself. I'm not going to require any of you to know this structure. But here's guanosine, and here is the 3-phosphates. So when GTPase comes in and hydrolyzes GTP to GDP, the GTPase specifically recognizes this third, it's called the gamma phosphate, okay? So it recognizes this phosphodiester bond between the second and third phosphates, and it removes the third phosphate and produces GDP. So what we have here in GTP gamma S is that one of the oxygens here on the third phosphate has been replaced by sulfur. Now sulfur is a big atom compared to oxygen, much, much bigger. So basically it produces a steric hindrance here so that when the GTPase comes in to hydrolyze this, it can't do it anymore because it can't recognize this phosphodiester bond between the second and third phosphate. So this is a stable form of GTP. And so imagine what happens here if we have this cycle going on, and instead of having GTP present, we have GTP gamma S. The GTP gamma S comes on, it activates the receptor, activates the effector, but then it stays activated, okay? Because GTPase cannot act on the GTP gamma S, and you've got a very long duration activation. It's not irreversible, because the GTP gamma S will eventually come off, but it certainly will be a much longer form of duration. But even more importantly, if this GTP gamma S is labeled with S35, we can now assay this entire cycle by the ability of the S35 to be activated. And this is a scattered plot which illustrates this activation, these two different states of the G protein, the G alpha subunit. So the basal state for GTP, for binding of the G alpha subunit to GTP gamma S is represented by these dark circles here. And this is a shallow scattered plot that indicates that this is a low affinity for GTP gamma S. So as long as the G protein is present in its basal state or its inactivated state, it has a low affinity for GTP or GTP gamma S. The affinity, the KD of this is about micromolar, 10 to the minus 6 molar. All right? So when you add agonists, and this is an experiment using an opiate agonist, a mu opiate agonist called Damgo. Once again, the agonist comes into the system, activates that whole cycle, and it switches the G alpha from a low affinity state into a high affinity state. So now you see this high affinity binding for S35 GTP gamma S. This is nanomolar label. So it's somewhere between 100 and 300-fold increase in affinity for GTP gamma S that you can measure. This is a huge increase, okay? And there's a lot of energy involved in that, in that process. And so what you're seeing here in this scattered plot is this switch coming from low affinity into high affinity, and that is the energy that drives the entire signal transduction process throughout the entire cell. All of the enzymes, the kinases, the ion channels, everything that's activated by these drugs all comes from this very fundamental shift from a low affinity state into a high affinity state for GTP. And you can see this here where you can actually look at different drugs and their ability to activate GTP gamma S binding. So once again, these are all opiates. And this compound here is a full agonist, and it produces full activation of this pathway. Other drugs like fentanyl and morphine are partial agonists. Buprenorphine is is a very low efficacy partial agonist. So in the clinic, these drugs show up as partial agonists, partial efficacy as analgesics. And you can see that this partial agonism is actually created at the very first step of signal transduction. It doesn't occur way downstream at some kinase level or something like that. Whether a drug is going to be a full agonist or a partial agonist or a full antagonist, that's all determined right here at the initial activation of the G protein transducing element. Skip this. Finally, in this process, you can actually utilize this as an autoradiographic technique. Now, remember last week we talked about using radioligand binding assays to localize receptors in their different areas of brain, and we talked about just using a standard radioligand binding assay to do this. Now, you can use S35GTP gamma S to do the same thing, but it's giving you very different kind of information. So here is here's a variety of different receptors that are activated by GTP gamma S autoradiographies. Here's cannabinoids, and you can see that the receptors are so the red color means that there's a high level of activation, blue means there's virtually none, and green means middle. And you can see, very high activation in some of the motor areas. Here in GABA B, which is the inhibitory receptor, a lot of cortical areas as well as midbrain. All these other receptors each one is different depending upon the localization of what's going on. Now, last week, the autoradiography that I was showing you was simply showing you where the receptors are. So the radioligand binding assay tells you nothing about the function of the receptors. Okay, they show you where the receptors are located, and you can infer function from that localization. But direct function is not here. But in this particular case, you're not just looking at where the receptors are, you're looking at where the receptors are activated by the agonist. Because if you don't, these are all studies were done where the slices are exposed to agonists for each individual receptor. So if you don't have any agonist at all, you see no activation. There's nothing there. You only see the binding when you throw in an agonist, and you get this G protein cycle, going over and over again. So this is showing you the localization of where the receptor function is really going on. Okay, this is showing you where receptors that are activated by agonists produce their effects. So it's a very different kind of autoradiography. Even though it looks kind of the same, it's giving you very different kind of information because it's more functional information than chemical information. Okay, all right. So that's a survey of the G protein. So on Friday, we're going to go past this into the other two components of the ternary complex system. So we'll talk about the receptors, and then we'll talk a little bit about the effectors, because I don't have time to go into that in detail. Questions about this? Okay, so don't forget your homework. If you have questions, just give me an email, and be happy to help you. If I'm around, I'm out of town the next 2 days. All right.
No.
Friendship is good.
Friendship.